of course, autograft does have some advantages. So it's from the patient, so it's not immunogenic. It won't cause an immune response. And it is biologic. It's, of course, a nerve, so it has the native structure. And it contains cells, especially the Schwann cells. Typically, that's what surgeons who are very much into using autograft really like to use autograft because it has Schwann cells. In addition, it provides attention-free repair, as we just talked about, and a lot of surgeons consider it to be free. There's no cost associated with it. But we know that autograft is not really free because the surgeon is creating a new nerve injury at the harvest site that comes along with a lot of potential problems. Like you can see in the pictures here, it can cause things like neuroma, or because you have a second surgical site, you have an additional site that has the potential for infection and dehiscence. It also requires some additional OR time. So you have to harvest the nerve, close the wound, and then construct a cable graft. So it does take some additional time. There's also finite nerve availability. You don't have an unending supply of nerve that you can take from. There may be inconsistent quality. You can't really choose the quality of the nerve that you're using. Sometimes the sural nerve may look, fairly decent and be round. Other times it can be really flat and ribbony. So you never really know what you're going to get when you pull an autograft. Again, there's the potential for donor site morbidity. There's limited regenerative space. And even though there are cells in the graft, there may be a questionable number of viable cells. And we're going to talk about that more in a minute. We're going to go through each one of these in detail. So what you'll see on the next slide are the two different ways that autograft can be harvested. And this is obviously for a sural nerve autograft. So it can be done open, where the entire leg is opened and the nerve is removed, which is what you see in picture A, which leaves a very large scar along the leg. Or it can be done in sort of a minimally invasive procedure where they just cut a small incision and then use a vein stripper to pull the nerve out. And sometimes they'll do this with multiple small incisions to sort of help remove it. And then you can see in the bottom picture what it looks like once they've pulled it out. There is a lot of donor site morbidity that goes along with removing an autograft. And you can see, as we discussed, there are a lot of different types of complications that can happen, and these things include neuroma formation, which then, of course, can cause pain. There is numbness from removal of the nerve. And again, the second surgical site adds potential for infection. And there was a study done in 2006 that showed that the sural nerve harvest complication rate is approximately 22%, which is pretty high. So the next thing we're going to talk about is the limited regenerative space. So when we talk about limited regenerative space, what we're talking about is the fascicular area that's contained in the graft. So when you look at the pictures here, what you see is everything that on the left-hand side, everything that's stained brown, those large brown circles, are all fascicles. And so when you look at the ulnar nerve, you can see that the fascicles are pretty big. There's a lot of them, and they're packed pretty closely together because that nerve doesn't have a lot of connective tissue in between the fascicles. So that the ulnar nerve and the median nerve both have about 50% fascicular area. So about 50% of that nerve is made up of fascicles. And if you remember back from your primary one, your axons are only going to regenerate through the endoneurial tubes in the fascicles. Axons will not regenerate through connective tissue. When you look at the sural nerve, what you see is that those fascicles are spaced much more widely apart, and there's a lot of connective tissue that's in between those fascicles. And the sural nerve only has about 33% fascicular area, or we like to call it regenerative scaffold because that's where regeneration is going to occur. When you look at the right-hand side of this slide, what that is showing you is two pieces of sural nerve put together as they would be in a cable graft. And then it shows it overlaid onto the ulnar nerve. And so you can see there's a lot of places where black is showing through that is not overlaid by the brown of the ulnar nerve. And so what that means is regeneration coming through those black fascicles would not grow into anything. They would be growing into connective tissue, and they would the axons would blindly end in that connective tissue. So when you're cable grafted in this manner, you may not be getting as much axon regeneration through the nerve as you could with a nerve that more closely approximated the fascicular area of the nerve that you were trying to repair. The next thing that we're going to talk about is this idea of cell viability. So autograft is very attractive to a lot of people because of there being Schwann cells contained within the graft. But this was a study done back in 2012 that actually looked at Schwann cell viability from nerves. And what they did was they took segments of nerve and they put them into different storage conditions. And different temperatures. So they did cold, they did warm, and they did room temperature. And you'll see that the different types of solutions that they put them in, one of them was sodium chloride, which is your typical saline. And then they did a couple of different types of basically cell culture they're solutions that people use for culturing cells in cell culture. So they're specifically meant to help keep cells alive. And this was an assay where they added a dye into the culture of the nerve, and that dye is taken up by the cells and it's fluorescent. And so there's an absorption spectrum. And so that's what they're showing here. And that tells them that that dye will only be taken up by the cells if the cell is alive. If the cell is dead, it will not be taken up by the cell. So it's a way that they can measure how many cells are still alive in that graft. And so you can see that very quickly, within the first hour, there is a large die-off of cells within that graft. And it happened most quickly and most precipitously for the grafts that were stored in the sodium chloride, which is, again, saline, the saline solution. So depending on how long it takes them to implant the graft after it's been removed, there really may not be as many viable Schwann cells in that graft as they think there are. And viability of Schwann cells is affected by a number of different issues. So once you devitalize it and remove the blood supply, of course, cells start to die. Again, time is a huge factor. So the longer you wait, the more cells die off. And they die off because there's a lack of oxygen and a lack of nutrients. And they also die off as other cells around them die off. So when you have a lot of cell death occurring in one area, they give off factors that can then induce other cells to also die off. And of course, the local environment also plays a role.